Good afternoon, all of you. Today we will see the next point, that is viruses. First, we'll see the introduction about viruses. So, what is viruses? The viruses is a nucleoprotein entity. Yes, these are viruses are the nucleoprotein entity which is able to utilize the machinery of another living cell for its multiplication. Yes, the viruses is a nucleoprotein entity which is able to utilize the machinery of another living cell for its multiplication. The name viruses means venom or the poisonous fluid. Yes, the name viruses, it means the venom or the poisonous fluid. The study of viruses is called as what? That is virology. The study of viruses is called as virology. The viruses and the bacteriophages are much smaller than the bacteria. Yes, what I'm saying? The viruses and what? The bacteriophages are the much smaller than the bacteria. They are made up of one of the nucleic acid, DNA or the RNA, or never both, and the protein. And the genetic material is enclosed within the protective coat of protein. The genetic material of what? The genetic material of viruses is enclosed within the protective coat of protein. In viruses, it causes the diseases like mumps, smallpox, rabies, influenza, or the AIDS. Yes, for example, the viruses causes different diseases like mumps, then smallpox, then herpes, influenza, or the AIDS. Then, so this is the introductory part about viruses. Then we will see the components of viruses. Viruses mostly consist of two parts, that is first nuclear, that is genome, and second, that is capsid. First part, that is nucleoid, or it is also called as genome, and second, that is capsid. An envelope is present in some viruses. A few enzymes are also known to occur occasionally inside the viruses. Here we can see the structure of virus. It shows presence of what? The matrix protein, the nucleocapsid, lipid envelope, and glycoprotein spikes. Yes, this virus shows presence of what? First, that is matrix protein, then nucleocapsid, then lipid envelope, and glycoprotein spikes. We will see one by one. First, nucleoid. It represents the viral chromosome. Then, the nucleic acid is the DNA or the RNA, but never both. Then, the DNA containing viruses is called as deoxyviruses. It means if the virus containing DNA, then it is called as deoxyviruses. And if RNA containing viruses, or if the viruses containing RNA, that is, it is called as riboviruses. As you know the meaning of DNA and RNA, ribonucleic acid and the deoxynucleic acid. Each of the viruses have two subtypes, double stranded and single stranded. So this is the nucleoid. Then second, that is capsid. So it is the proteinous covering around the viruses which protects the genetic material. Yes, it is the proteinous covering around the viruses which protect the genetic material from physical and chemical agents. Yes, this capsid, it protects the genetic material from what? From the physical and the various chemical agents. Then the capsid consists of number of subunits, and these subunits are called as capsomers, or the this subunit is called as capsomers. Then in some animal viruses there is a loose membrane covering. Yes, in some animals there is loose membrane covering that occur in some animal viruses, and rarely in plant and bacterial viruses. Then the viruses without an envelope is called as naked viruses. Yes, here I am repeating. I am saying the viruses without envelope is called as naked viruses or naked virus. Then third, envelope. The some viruses, here you can see the structure. Some viruses contain phospholipid bilayer known as envelope. So what is envelope? Some viruses contain phospholipid bilayer, and this phospholipid bilayer is called as envelope. Then the virus lacking envelope is called as naked viruses, or the without envelope is called as naked virus. Then envelope is a lipid bilayer which is acquired from the host cell membrane. Here we can see the structure of envelope or location of envelope. The envelope is a lipid bilayer which is acquired from the host cell membrane. Then we will see the general properties of viruses. First property, they are or the viruses are ultra microscopic infectious particle having size or the having diameter 20 to 350 nanometer. Then second, they are nucleoproteins. Third, they contain either DNA or the RNA, which is either single or the double stranded. Yes, they contain what? They contain either DNA or the RNA, which is either single or the double stranded. Then DNA or the RNA has protein coat to more complicated structure. This DNA or the RNA having has a coat of protein or the protein coat to more complicated structure. Then they are highly pathogenic. Next, they lack protoplasm. Then they grow and undergo the multiplication and mutation within living host cell. Then they infect the host by nucleic acid. Then they easily spread from one organism to another. Then they easily spread from one organism to another. Then they are resistant to antibiotics. Then they can be crystallized. Then they are obligated intracellular parasite, that is they produce and develop on host cell of animals, plants or the bacteria. Then outside the host cell, the viruses exist as individual particles, and these individual particles is called as virions. Yes, keep in mind it's important. These individual particles are called as what? Virions or virions. Then based on the above mentioned property, it cannot be prove that whether viruses are living or the non-living, since they possess properties of both. First we will see the properties or the characters of non-living being. First, they lack protoplasm. Then they lack enzyme system. First character, they lack protoplasm. Then next, they lack enzyme system. Then they do not respire. Next, they can be crystallized. And last, they cannot be cultured outside the cell even by using culture media. Then characters of living being, then in the first, they can undergo replication within the living cell. The nucleic acid present in them synthesize the protein for their coat, although they utilize host rhizome for protein synthesis. Then they cause a diseases. Then gene mutation in nucleic acid. The gene mutation in nucleic acid are similar to that in the chromosomes of the living organism. So this is all about what? The characteristics or the general properties of viruses. Next, that is characteristics of viruses. We will see the different characteristics. First, that is virus size, then shape, then symmetry, viral replication, metabolism and resistance. In resistance, the temperature, cold, radiation, organic solvents and disinfectant. First, that is viral size. The size of viruses or the viruses are smallest disease causing agent in, these are the smallest disease causing agent in living organism. The plant viruses ranges in size from 17 nanometer to 2000 nanometer, and while the animal viruses having the size 20 to 350 nanometer. The plant viruses having size 17 nanometer to 2000 nanometer, and the animal viruses having the size 20 to 350 nanometer. Then viruses shape. Here we can see the different shapes of viruses. The shape of viruses is greatly varies, or they are varies. For example, rod shaped or the filamentous, for example TMV. Then brick shaped viruses, for example pox virus. Then bullet shaped virus, for example rabidoviruses or the rabies virus. Then spherical shaped viruses, for example HIV. Then another example of spherical shaped viruses, influenza and herpes viruses. Then next, tadpole shaped viruses. The example of this viruses, that is bacteriophages. These are the few shapes of viruses along with their examples. Here we can see the different shapes of viruses along with examples. Then next, symmetry. Here we can see the symmetry of these viruses. First TMV virus, then polio, and third, that is bacteriophages. In TMV viruses it shows capsomer, then capsid and genome. And this capsid and genome is called as nucleocapsid. In case of polio virus, it shows presence of protein coat, then nucleic acid inside the coat, and isometric and neck. It is not envelope. And in case of bacteriophages, it shows presence of protein coat, tail, and the tail fibers. It is symmetry. In case of TME virus, it is helical symmetry. In case of polio virus, it is polyhedral symmetry. And in case of bacteriophages, it is complex symmetry. It means TME virus, B it is polyhedral, and C it means complex symmetry of C it means bacteriophage. The morphological protein subunit of capsid are arranged together to form a symmetrical structure of the viruses. The two basic symmetry are recognized in viruses, and they are helical symmetry and the icosahedral symmetry. In some viruses, symmetry is more complex, which is another than helical or the icosahedral symmetry. Yes, I am repeating here what I am saying. In some viruses, the symmetry is complex. It means which is other than what? The helical symmetry or the icosahedral symmetry. Then next, that is viral replication. The viruses only replicate inside the host cell. The viruses only replicates inside the host cell. Then metabolism. The viruses are metabolized inert. The viruses are metabolically inert outside host cell. Then they are also called as obligate intracellular parasite. Then last characteristic, that is resistance in that first temperature. The most viruses are heat labile. Then the viruses are inactivated by heating at 60 degrees Celsius for 30 minutes or the 100 degrees Celsius for few seconds. Yes, in temperature, first the most viruses are heat labile, and second the viruses are inactivated by heating at 60 degrees Celsius for 30 minutes or the 100 degrees Celsius for few seconds. Then cold. Viruses are stable and resistant to cooling. Then the virus can be stored for long duration. The virus can be stored for long duration at minus 40 to minus 70 degrees Celsius by lyophilization or by freeze drying. Then next, radiation. Both ionizing radiation as well as non-ionizing radiation can kill the viruses. Yes, both ionizing radiation as well as non-ionizing radiation can kill the virus. Then UV rays causes pyrimidine dimer formation, while ionizing radiation, for example X-ray, causes the lethal break of viral genome. Then next, organic solvent. The chloroform, ether and bile salt can destroy the all viruses by lipid solubilization. Yes, in organic solvent, for example chloroform, ether and the bile salt can destroy the all viruses by lipid solubilization. Then disinfectant. Most viruses are destroyed by oxidizing agent, for example chlorine, H2O2 or the iodine. Yes, most of the viruses are destroyed by what? Destroyed by destroyed by oxidizing agent, for example such as chlorine, H2O2 or the iodine. Then many viruses are resistant to phenol and the chlorination. Yes, most of the viruses are resistant to phenol and the chlorination. The phenol and chlorine do not always inactivate the enterovirus, particularly if they are present in fecal material. Yes, when that is if they are present in particularly if they are present in fecal materials. Then last, that is antibiotics. Viruses are resistant to antibiotics. So this is all about the introduction, then components of the structure of virus, general properties and the character or the different characteristics of viruses. I will stop here. Next part we will see in next lecture.